think we should be good to start. Hello, good afternoon, everyone. I'm Sophia Foch, Sales Associate or Cryo-Free Crusader, according to my email signature, here at Atelerix. And I'll be hosting this webinar today, along with our presenter and CSO and co-founder, Dr. Steve Swioklo. Our webinar title today is Future-Proofing Advanced Cell Models, the Logistics Revolution. And the presentation will be around 20 to 25 minutes, followed by a Q&A session. Feel free to drop any questions you have into the Q&A box below, and we can get those answered for you after the presentation. So over to you, Steve.
Thank you very much, Sophia. Thank you all for joining this webinar. This is really going to introduce our technology and its applications in advanced cell models. As everyone's aware, advancements in cell-based models have significantly enhanced our ability to study human diseases and test new drugs, offering real alternatives to traditional animal testing. These advanced models, which often sit under the umbrella of New Approach Methodologies, or NAMs, include spheroids and organoids, bioprinted or engineered tissues, scaffold-based or stratified models, and organ-on-a-chip or microfluidic systems. And what these advanced cell models aim to do is to recapitulate the complex 3D architecture and cell behavior of tissue and organ systems to allow for physiological relevant and disease-specific systems for assessing new drugs. And the use of NAMs are increasingly encouraged in drug development and IND submission. And this is supported by the FDA Modernization Act and new pilot programs that are really encouraging the use of new approach methodologies, these advanced models, to replace or run alongside animal models. But the problem with these models really is that as cell model complexity and physiological relevancy increases, the time and expertise required to generate reliable cell models also increases. Essentially, they're a lot more difficult to culture and form. So a possible answer to this is to generate these cell models at sites of excellence, where there's the equipment and the expertise, and then send these directly to sites for drug development. But the problem with this is that the models are complex. The more complex they become, the more difficult they are to freeze, and they won't survive at room temperature. Their complexity also leads to high fragility, and often they're too fragile to ship. So really you're stuck between a rock and a hard place. But we're here to say that it doesn't have to be this way. We have a platform technology that enables the shipment of these complex cell models at room temperature. And I'll describe really how the products are used. But first, I really want to just start with what goes wrong at room temperature. So room temperature preservation, also known as hypothermic preservation, is really quite a simple process. At 37 degrees or normothermia, so the temperature in your body or in the incubator, cells are metabolically active. They've got high enzymatic activity and they're running multiple different cellular processes. The idea behind hypothermic storage is that as you decrease the temperature to hypothermic temperatures, that's defined as anything below 32 degrees, the enzymatic and metabolic activity slows to such an extent that cells are paused. They're in a state of suspended animation. They don't really have the energy to fulfill any of their cellular processes. So they are in a state that they can theoretically be stored. But what happens also when you decrease temperature is that at 37 degrees, cells have an active fluid membrane, but at these lower temperatures, there's a phase change in the membrane state that essentially makes them static. The lipid membranes of these cells become static. It's a little bit like if you think about butter or fats, lipid membranes are full of fats and they become rigid. And it's this rigidity that causes pores to form in the cell membrane. And this results in uncontrolled ion and water entry into the cell. And it's this dysregulated movement of components into the cell which causes continual membrane destruction, edema, and eventual cell death. And this is the basic first mechanism, which is the lead cause that results in cells not being able to survive outside the incubator. They may survive a matter of minutes, matter of hours, but it's not very long. So to prevent this, we have a technology that involves alginate hydrogels that encapsulate and protect those cells. Alginates are naturally occurring polysaccharides which are derived from brown seaweed. They form a hydrogel when cross-linked, and they're biologically inert, non-toxic, and biodegradable. They're very versatile, which means that they can be cross-linked very easily. So what that means is being turned from a liquid gel to a solid gel. And they can also be dissolved very easily. So they can be returned back to a fluid state. So can be used very simply with cell procedures and SOPs. They're also xeno-free and protein-free. And we use specific formulations of alginates that offer maximal cytoprotection during hypothermic storage. In essence, the gels stabilize cell membrane integrity and also prevent the uncontrolled transport of ions and water into the cell. And this prevents that continual destruction that I mentioned in the earlier slides, and means that the cells themselves can be stored at extended periods. It applies to fresh cells, cell models, tissues, and viruses. And rather than days, they can be stored for up to 2 weeks without the need to freeze. And we have also formulated these hydrogels to offer maximal mechanical protection during shipping. So the fragility of these models mentioned earlier means that you need a gel that has suitable mechanical properties to prevent shearing from liquid movement and also buffer mechanical forces. So these are vibrational and drop and shock forces that you would often see during shipping. And we supply these gels in very, very easy to use kits. They are fully packaged to allow a very simple encapsulation of your samples. And what these kits do, they extend the preservation and maintain sample viability and functional performance. And what they do, they enable complex tissues, bloods, sensitive cells, all the way down to therapies. They allow them to be shipped. They're easy to validate. They de-escalate shipping. What this means is that they're very easy to use. And they can slash costs for by up to 10-fold compared to either expedited shipment or cryo shipment. And what this means is they offer a flexible, low-cost supply chain and workflow from sample collection to final application. And these have also been developed to be suitable for multiple different cellular formats or biosample formats, which really offers you a toolkit. Cytostore is used for cells, viruses, and also mRNA LNPs. Well-Ready is specifically used for in-plate models. That might be model layers or organoid models or culture insert models, covers the whole range. Tissue Ready is suited for tissues and organoids for preservation in a vial, and Tissue Ready Plus for tissues. And we also have products for blood products, Blood Ready for whole blood, and Leukostore for leukapheresis. So we have multiple products that are used throughout customer workflows or drug development workflows. And I just wanted to talk through a couple of examples of these workflows, focusing on organoids as a really probably the most widely used, increasingly used 3D model currently for animal replacement. And the first example that I wanted to talk about was patient-derived tumor organoids. These are used very commonly in cancer drug development. And to make these organoids, you take tissue. It's either a cancer resection or a biopsy. You mince this tissue, dissociate it, and derive the cancer-positive cells, normal cells, and also cells of the microenvironment, the tumor microenvironment. These cells are initiated to become organoids by embedding them, usually within a gel. And then you have organoid formation, expansion, and passaging over a period of time, and eventually QC validation, and then you can perform your assays. And these assays are commonly disease modeling, like looking at cancer progression, drug screening, like personalized chemotherapy, and also toxicity testing. And throughout this process, there are a number of points that really cause a little bit of a logistical nightmare. And the first is actually right at the start with tissue. So tissue requisition in itself is very, very tight, inflexible, and resource heavy. It's hard to predict when a patient is going to be ready for surgery. When the patient is ready for surgery, you've got to very rapidly coordinate couriers to pick up that tissue. And then when it arrives at the site of processing, it needs to be processed straight away. If it arrives on a Friday, people need to stay late on a Friday, work into the weekend, etc. And this is because most tissues only have a shelf life of 24 hours. Beyond that, you've got a rapid decline in the viable cell recovery. And we developed Tissue Ready Plus to avoid this decline. Not only does Tissue Ready Plus preserve histological integrity and staining profiles, which are very useful in certain models like precision cut slice models. Importantly, the product maintains the recovery of viable cells. So there's a very small graph that you can see there that shows that those tissues preserved in Tissue Ready Plus in the orange bar maintain close to 100% of viable cancer cells and also normal cells after 5 days of storage. But if this was going to just be stored in medium, in the bluey teal bar, you get a rapid decline in viable cell recovery. So it's really not suitable, and Tissue Ready Plus preserves the phenotypically normal cells that you need from cancer tissue. Often alongside tissue, patient-matched blood is collected. Patient-matched blood is often used for immunocompetent models. Essentially, the immune cells are taken from the blood and then co-cultured with organoids or tissue. We also have a product for whole blood, mentioned earlier. And really what Blood Ready is able to do, that's the product. It's able to preserve those very fragile subpopulations of major lymphocytes populations, and also preserve the epitopes on those cells. So Blood Ready can be used in line with Tissue Ready to enable the shipment of patient-matched blood. Just as a CytoStore can also be used to ship dissociated cells. Sometimes it's useful if there is a site of collection near a hospital, for example, where there's expertise to dissociate those cells can be dissociated and then sent on to sites where organoid generation can be carried out. It's also important just to talk about how long this process takes. So Organoid generation from tumors is a long and highly skilled process. It can take 3 to 5 weeks. And there are many points where failure can occur. It could be in dissociation, embedding, contamination, because that culture protocol is so long. So in many scenarios, centralized manufacture and distribution of these models enables the QC and release from validated organoid generation processes to be sent to sites of use. And this aids in uniformity and reproducibility of these models and the results that you get from these models. So to do that, you need to be able to ship the assays or the organoid that forms the assay. And here's just a quick example of some of the work that's been done with cancer organoids, breast and lung. And these organoids were stored for up to 7 days with no reduction in yield, viability, or growth. And what this enables is this long process of organoid generation to be carried out in sites of excellence, and those organoids can then be further shipped on. So essentially, we enable the shipment of live assay-ready patient-derived organoids in vials or plates. The second example I wanted to give was iPSC-derived organoids. So in this process, iPSCs are expanded and characterized, and then they're differentiated into the 3 germ layers. After this occurs, organoid initiation and embedding takes place, and then organoid maturation and patterning. That is before QC validation and assays are carried out, similar to the other organoids discussed. And these assays are commonly disease modeling, such as modeling developmental disorders, drug screening and toxicity testing, and also gene editing studies. And with iPSC-derived organoids, it can be an exceptionally long culture process to derive fully mature organoids. This can be as much as 18 weeks, 4 to 18 weeks, it says there. So that's a very, very long period where multiple things can go wrong. And you need a highly skilled, resource-heavy, it's highly skilled, resource-heavy with a high risk of failure. So really, it's quite high risk to actually get from your iPSCs to a final product which can be used for screening. And again, this is where centralized manufacture and distribution comes in as a possible option. People with those expertise can actually culture these organoids within a uniform process and then ship them out. And we worked with a number of different iPSC-derived products. And this is just an example of iPSC-derived liver organoids, which were preserved in plate with well-ready. And as you can see, the viability, structure, and phenotype of these products were preserved after up to 5 days storage in well-ready at room temperature. And importantly, these organoids retained liver metabolic function up to the level of pre or non-stored fresh organoids, so are suitable for hepatic screening. So, as with the cancer organoids, our products enable the shipment of live assay-ready iPSC-derived organoids in either vial format or plate format. And just as a side, CytoStore can also be used to ship iPSCs. So if people have iPSCs, disease-specific iPSCs, or specially generated iPSCs, these can also be shipped to collaborators using our products. So the capabilities of the technology extend across multiple advanced models. To date, as we know of, the products are being tested on a range of different spheroids and organoids. These include lung cancer, CRC, colorectal cancer, breast cancer, iPSC-derived brain, cardiac, dermal, liver, and lung organoids. They've also been used to preserve bioprinted engineered tissues. These include iPSC-derived and MSC-derived bioprinted models, cardiac and skeletal engineered microtissues, and iPSC-derived cardiomyocyte printed models. And they've also been used to preserve scaffold-based or stratified models, so skin, gut, airway models that are cultured within cell culture inserts. And this list is always expanding. We're always having customers preserving more and more different advanced models, really increasing the validation across different screening modalities. And the advantages of our products extend across multiple industries. Suppliers are able to supply ready-to-use models rather than sell cells and accessories. So accessories are plates, medium, etc., for users to culture themselves. What this allows them to do is to supply a higher value offering, reduce failure rate due to error or contamination, and also increase reordering rate as the products work. They're readily QC'd and people can just use them in an assay-ready fashion. Also, CROs, they're now able to ship models directly to their sponsors. This is often useful to enable confirmatory testing on the exact same model where screening is being carried out within that CRO. And also allow the screening of late-stage development drug candidates that may not be able to be released from, say, pharmaceutical sponsors because of their late stage of development. They can be used by pharma and biotech to share models between multiple sites and also pause workflows. This enables them to share models with sites that have different expertise, pause culture workflows to increase resource flexibility. Essentially, they can use our products to stop the process of culture in case there is a requirement of low resource or a holiday that prevents that culture process to carry on. And they can also access a greater range of models. Some of these are coming from the suppliers themselves. Being able to share these models really increases accessibility. And all of these sectors benefit from the reduction on reliance on animal models. The enablement of shipping models that have gone through a process that results in them being very, very well validated, increases the availability of harmonized models for consistent testing. What that means is that multiple sites with multiple programs and different expertise can have access to the same models to enable the improved multi-center testing for drug discovery. There's an enhanced supply chain flexibility for human tissues and blood. We talked about that. Obviously, often the geographical limitations of sourcing tissue and blood cause a lot of problems, but this can be enhanced. And also increase the accessibility of these models for wider use. They're no longer just privileged to the sites that people can make them. So that's all good for science and good for animal welfare. And with that, I'll just end the presentation and hand back to Sophia. And Sophia will open up to any questions that you may have. Thank you.
Thank you, Steve, for that fantastic and informative presentation. You battled through your cold well there.
Thank you.
On to the Q&A and for Steve to answer some of your submitted questions. So do feel free to continue sending them through and we can get them answered in this time. So the first one is, has Well Ready been used to ship plates internationally?
Yes, it has. Yeah, it's successfully been used for shipment of products from UK to US and internationally, actually. The shipment of plates on airline travel has been pretty successful.
Thank you. Could you explain more on the in vitro toxicity performed with the hydrogel encapsulation? So have you conducted standard toxicity assays such as MTT, ATP on cells after release from the gel?
Yes, so those sort of standard outputs have been employed by ourselves in internal development, but also collaborative partners and customers using our products. We've got a data pack that kind of lists all of the outputs of these commonly used toxicity assays, and they kind of range quite widely from simple MTT tests all the way up to quite complex kinase binding assays and that sort of thing.
Thank you. And they further asked if we can share publications that detail Atelerix's studies on the toxicological safety, particularly in the context of cell-based assays for drug discovery or clinical applications as well.
Yeah, excellent. We'll be more than happy to share those after this presentation.
Thank you. Are any of the products CGMP compatible?
All of the components within the products are from GMP compliant sources. Our manufacturer is not GMP compliant. We operate to ISO 9001. But for standard use within preclinical discovery, they don't necessarily need to be CGMP compliant. Our quality management system is fully auditable by users of our products.
Thank you. Is there scope to develop the technology further? For example, to be able to store tissue for long-term storage in place of freezing?
There may be scope. There are complexities with actually the long-term storage of tissues. They will have a finite lifetime always. I mean, they will start to degrade. What our technology does is slow down essentially this process of degradation to extend the fresh shelf life. There are potential opportunities that the gel could be used in combination with freezing, but this also comes with a number of different complexities, mostly due to the shock incurred by the freeze-thaw process. But there's always scope for further development.
Great. Have your alginate hydrogels been tested for endotoxin?
They are tested for endotoxin, yes, and there's a lower limit for release.
We have a question further on from the previous one asked. Are there any plans for CGMP production for future use in clinical trials?
Not immediately. We do actually work in with clinical trials for the provision of our products for cell-based therapies. Actually, it's less of the model, and what we're able to do is transfer manufacturing to sponsors so that they can actually do the reformulation of CGMP compliant products within their own process. And we've done that successfully for clinical trial partners. We won't necessarily start manufacturing ourselves. We have other plans that we can also use contract manufacturers to manufacture for us under a GMP compliant manufacturing system. So there are a number of options for us, but we won't be doing it ourselves.
Thank you. Does it work on 2D models as well as 3D ones?
It does, yes. So it was initially designed for 2D models, anything from iPSC derived cortical neurons to DRG neurons to hepatocyte models to different somatic cells. It's been used for a number of different 2D models. Obviously, the complexity of 2D models is reduced, but they're still used widely, especially iPSC disease relevant model layer models, which people use our product for quite regularly.
Thank you. Is any specific temperature control needed?
Yeah. So we advise shipping products, shipping cells or cell-based products using our technology. We advise keeping the temperature between 15 and 25 degrees. And so when we're talking about international transport, so transport on airlines, we advise the use of controlled room temperature boxes, which are just small polystyrene boxes with phase control packs, phase change temperature packs in. They're available on our website and they're very widely available. They're generically used for the shipment of pharmaceuticals and just allows the shipment at room temperature without any massive deviations in that temperature.
Thank you. And further on from that, is shipping at room temperature, or how is shipping at room temperature more environmentally friendly than shipping on dry ice?
Shipping at room temperature allows you to massively reduce the volumetric weight of shipments. So you don't need a huge polystyrene box with 20 kilograms of dry ice in to ensure international shipments. And dry ice in itself is very energy intensive to produce. And the freight weight of that large package is very heavy, induces a lot of extra fuel, and is less energy efficient to send. So we've calculated in some instances that shipping using our product compared to traditional cryopreservation approaches can reduce carbon emissions by, I think it's up to about 98%. It's 97.7% or something. So there can be a big carbon efficiency saving.
Yes, thank you very much. What are your main competitors in tumor-derived organoids, and what are our advantages over that?
Yeah, so in terms of competitors, some tumor-derived organoids can be frozen. It's still a relatively limited range due to the complexity of freezing those tumor organoids, but that's not to say it can't be done. So that's the main competitor, really, the cryopreservation of tumor organoids. But as the complexity of these cell models increase and the phenotypic and physiological relevancy, as I said at the start, the ability to freeze these just reduces and reduces. So there'll be more of a demand for non-frozen approaches as time goes along.
Thank you. Are processes for encapsulation automatable?
Yeah, they are. So they can be handled using very simple liquid handling processes. So it's fully automatable and scalable.
Thank you. And are your products xeno-free?
Yes, they are. Yes, they're completely xeno-free, protein-free, free of any animal bits or anything else.
Thank you. And what is the shelf life of these kits?
Shelf life is up to 1 year, depending on the product.
Great. And I think we have one more. Are there any special equipment required for the protocols?
No, it's very straightforward. The protocols themselves, depending on the product, either just require a very quick decant process to encapsulate your sample. Some require pipettes, but nothing more elaborate than that. So they're very easy to use and can be very quickly adopted into people's processing.
Excellent. Thank you, Steve. If we don't have any more questions for now, I think we can draw that part to a close. If you do think of any more questions after the presentation, then please feel free to reach out to us in our sales@atelerix.co.uk inbox or reply to our follow-up emails after the webinar. So we have actually recorded this presentation. It will be available on our YouTube and socials if you'd like to recap anything we've discussed or share it with your colleagues or collaborators. If you are keen to know more or are looking to trial our products, then we're happy to organise an evaluation and you can give our hydrogel encapsulation method a real go. Thank you to everybody for joining the call and we'll catch you on the next one. Thank you, Steve.
Thank you. Goodbye.